A breakthrough in Alzheimer's prevention. The promise of early amyloid removal. The quest to defeat Alzheimer's disease has taken a significant step forward, with groundbreaking research suggesting that removing amyloid plaques from the brain decades before symptoms appear may prevent or delay the onset of this devastating condition. Published in the Lancet Neurology, the latest findings from the Knight family dominantly inherited Alzheimer Network Trials Unit, DAN2, represent what could be the first clinical evidence that early intervention targeting amyloid pathology can alter the course of Alzheimer's disease. A monumental achievement in the field of neurodegenerative research. This international study, led by researchers at Washington University School of Medicine in St. Louis, examined 73 individuals with rare inherited genetic mutations that virtually guarantee the development of Alzheimer's disease in middle age. Among a subgroup of 22 participants who had no cognitive problems at the study's inception and received treatment for an average of 8 years, the experimental drug gantenerumab reduced the risk of developing symptoms from essentially 100% to approximately 50%. A remarkable outcome that challenges our understanding of what's possible in Alzheimer's prevention. The amyloid hypothesis gains new validation. For decades, the scientific community has debated the role of amyloid plaques in Alzheimer's disease. The amyloid hypothesis posits that the accumulation of these protein deposits in the brain triggers a cascade of events leading to neurodegeneration and cognitive decline. Critics have long questioned whether amyloid represents the primary cause of Alzheimer's or merely a downstream effect of other pathological processes. This new research provides compelling evidence that removing amyloid plaques before symptoms manifest can indeed delay or prevent the onset of dementia, lending substantial support to the amyloid-first model of disease progression. Everyone in this study was destined to develop Alzheimer's disease, and some of them haven't yet, explained Dr. Randall J. Bateman, senior author of the study, and Charles F. and Joanne Knight, distinguished professor of neurology at WashU Medicine. We don't yet know how long they will remain symptom-free, maybe a few years or maybe decades. His words capture both the uncertainty and hope that define this research frontier. The study builds upon the Knight family DIAN-2-001, the world's first Alzheimer's prevention trial, which launched in 2012 with the ambitious goal of evaluating anti-amyloid drugs as preventive therapies. This pioneering effort specifically targeted individuals with dominantly inherited Alzheimer's disease, those whose genetic makeup virtually guarantees they will develop the condition, often at unusually young ages. These rare cases, while affecting only a small subset of Alzheimer's patients, provide an invaluable research model because of their predictable disease progression, from mixed results to meaningful success. The path to this breakthrough was not straightforward. When the original DIAN-2-001 trial concluded in 2020, results were mixed at best. While gantenerumab successfully lowered amyloid levels in the brain and improved some measures of Alzheimer's-related proteins, researchers did not initially observe cognitive benefits. This lack of apparent efficacy was particularly puzzling in the group without symptoms, regardless of whether they received the drug or placebo. The trial leaders faced a critical decision: abandon the pursuit of this therapeutic approach, or continue exploring whether higher doses or longer treatment duration might yield different results. Choosing the latter path, they launched an open-label extension study that would ultimately reveal the transformative potential of early amyloid removal. In the extension trial, all participants with high-risk Alzheimer's genetic mutations received gantenerumab, regardless of their treatment history in the original study. This design eliminated an internal control group, forcing researchers to compare results with data from the DIAN observational study, a related effort tracking individuals with Alzheimer's mutations who received no drug treatment, and with placebo-treated participants from the original trial who did not continue into the extension. The results from this extended treatment period were striking. Among participants who had no symptoms at baseline and received the longest treatment duration, averaging 8 years, the medication cut the risk of developing Alzheimer's symptoms by half. This 50% effect size represents a statistical calculation that accounts not only for how many people developed symptoms, but also when those symptoms emerged relative to their expected age of onset based on family history. The importance of treatment duration. One of the most significant insights from this research is the apparent necessity of sustained treatment for prevention. Participants who received gantenerumab only during the 2 to 3 year extension phase, because they had received another drug or placebo during the original trial, showed no observable effects on cognitive function. This contrast with the 8-year treatment group suggests that intervening years before expected symptom onset may be crucial for achieving preventive benefits. This finding has profound implications for how we think about Alzheimer's treatment timing. Traditional approaches have focused on treating patients after diagnosis, when significant neurological damage has already occurred. The DN2 results support a paradigm shift toward much earlier intervention, potentially beginning decades before symptoms appear. The dynamic nature of the measured effect size introduces both hope and uncertainty. As some participants are at or just past their expected age of onset, the longer they remain symptom-free, the more substantial the preventive effect becomes. Conversely, if currently healthy participants develop symptoms in the future, the effect size could diminish. This uncertainty underscores the importance of continued monitoring and research. Balancing benefits and risks. No medical intervention comes without potential side effects, and gantenerumab is no exception. The drug has been linked to amyloid-related imaging abnormalities, RIA, which manifest as tiny spots of blood in the brain or localized swelling. In the extension study, ARIA rates increased to 30% from 19% in the original trial, which researchers attribute to the higher doses used. Most cases of ARIA remain asymptomatic and resolve spontaneously. However, more severe cases can occur. In this study, 2 participants developed ARIA severe enough to require discontinuation of treatment, after which they recovered. No life-threatening adverse events or deaths were linked to ARIA, and the overall safety profile remained consistent with that observed in the original trial and other gantenerumab studies. The risk-benefit calculus for preventive Alzheimer's treatment differs fundamentally from that for symptomatic patients. Individuals with genetic certainty of developing the disease may be more willing to accept potential side effects in exchange for delaying or preventing cognitive decline. However, as we consider broader applications of this approach, careful consideration of these safety issues becomes paramount. From rare mutations to common disease. While the DIAN-2 study focused exclusively on individuals with rare genetic forms of Alzheimer's that cause early onset, researchers believe the findings will inform prevention and treatment strategies for all forms of the disease. Both early onset and late onset Alzheimer's share a common feature. Amyloid begins accumulating in the brain approximately 2 decades before memory and thinking problems emerge. This temporal pattern suggests that the window for preventive intervention may be similarly long in late-onset Alzheimer's, which affects the vast majority of patients. Furthermore, clinical trial results from early-onset Alzheimer's mutation families have historically been replicated in late-onset disease studies, providing additional confidence in the broader applicability of these findings. Dr. Bateman expressed optimism about the future. If late-onset Alzheimer's prevention trials have similar results to the DN2 trials, there soon could be Alzheimer's preventions available for the general population. I am highly optimistic now, as this could be the first clinical evidence of what will become preventions for people at risk for Alzheimer's disease. The road ahead. The discontinuation of gantenerumab development by Roche Genentech in November 2022 precipitated an unexpected change in the study's trajectory. This decision, based on disappointing results from Phase 3 trials in patients with early symptomatic Alzheimer's disease, forced researchers to adapt their ongoing work. Most participants in the international open-label extension have transitioned to receiving lecanemab, another anti-amyloid drug approved by the FDA in 2023 for slowing cognitive decline in patients who already exhibit Alzheimer's symptoms. This transition raises important questions about the consistency of preventive effects across different anti-amyloid agents. While gantenerumab and lecanemab target the same protein, their mechanisms of action and pharmacological properties differ. Continued research will determine whether the preventive benefits observed with gantenerumab can be maintained or enhanced with alternative therapies. The Knight family DIAN-2 has launched the amyloid removal trial to explore how long dementia can be delayed through amyloid-targeted interventions. Additionally, they are evaluating remternetug, an investigational amyloid-removing drug manufactured by Eli Lilly, in the primary prevention trial. This study represents an even more ambitious preventive approach, enrolling individuals as young as 18 with minimal detectable Alzheimer's-related molecular changes, up to 25 years before expected symptom onset. Broader implications for Alzheimer's research. The DN2 findings have catalyzed renewed enthusiasm for amyloid-targeted prevention strategies across the research community. Dr. Maria C. Carrillo, Alzheimer's Association Chief Science Officer, emphasized the significance of these results. These exciting preliminary findings hint very clearly at the potential role of lowering beta amyloid in prevention of Alzheimer's disease. The implications extend beyond individual therapies to reshape our fundamental approach to Alzheimer's disease. Rather than viewing it as an inevitable consequence of aging that can only be managed after onset, these results suggest we should conceptualize it as a preventable condition with a long pre-symptomatic phase amenable to intervention. This shift demands new frameworks for risk assessment, early detection, and preventive care. Identifying individuals at high risk for Alzheimer's decades before symptom onset becomes crucial, a challenge given the limitations of current biomarkers and the cost implications of widespread screening. Genetic testing for common Alzheimer's risk factors like APOE4 may become more prevalent, though the ethical and psychological considerations of providing such information require careful navigation. Challenges and opportunities. Despite the encouraging results, significant challenges remain in translating these findings into widespread clinical practice. The cost of anti-amyloid therapies, which can exceed $56,000 per year, raises questions of accessibility and healthcare system sustainability. Additionally, the long treatment duration required for preventive benefits, potentially decades, creates unprecedented demands on healthcare infrastructure and patient adherence. The identification of optimal treatment candidates presents another complex challenge. While genetic testing can identify individuals with dominantly inherited Alzheimer's, the broader population at risk for late-onset disease encompasses a more diverse group with varying degrees of risk. Developing effective risk stratification tools and determining appropriate intervention thresholds will be critical for implementing preventive strategies efficiently and ethically. Conclusion: a new era in Alzheimer's prevention. The DIAN-2 findings represent more than just another incremental advance in Alzheimer's research. They herald a potential paradigm shift in our approach to one of medicine's most formidable challenges. By demonstrating that early removal of amyloid plaques can delay or prevent the onset of dementia, this research provides the first clinical evidence that Alzheimer's disease may be preventable rather than merely manageable. As Dr. Bateman noted in his forward-looking assessment, "One day soon, we may be delaying the onset of Alzheimer's disease for millions." This vision, while still aspirational, now rests on a foundation of clinical evidence that was absent just a few years ago. The journey from these promising preliminary findings to widespread preventive treatment will require continued research, refinement of therapeutic approaches, and careful consideration of implementation challenges. However, the fundamental message remains clear. The long-held belief that Alzheimer's disease is an inevitable consequence of aging may soon be relegated to medical history. For the millions of people worldwide concerned about their cognitive future, these findings offer something truly transformative: hope. While many questions remain unanswered, the door has been opened to a future where Alzheimer's disease, rather than being an inexorable decline, becomes a condition that can be prevented through timely intervention. The promise of preserving cognitive health late into life moves from theoretical possibility to realistic expectation, marking the beginning of a new chapter in our fight against this devastating disease.